Hi students, this is my video. I'm going to explain the details to the chemical reaction, cellular respiration. Before you're watching this video, you should have already completed the study guide that goes with the textbook. If you haven't, stop now, go do that first because this is going to go into way more detail, so you need the overview that you get from the study guide. While you're watching this lecture video, you need to be taking notes. Like this, I'm going to be drawing things on the board and filling in this mitochondria diagram. So I want you to have this diagram and fill it out with me while I'm going over this. Of course, you can stop, pause, anything you need because it's a video. But I'm only going to explain things really once. So let's go ahead and get started. So this is a mitochondria, a really basic drawing of a mitochondria. This is where cellular respiration happens, and so if you've done the study guide, you should have seen the chemical reaction for cellular respiration, but it is glucose, C6H12O6, plus oxygen. So the food we eat, we get carbs, which is glucose is a carb, and then the oxygen we get from air we breathe. Those are the reactants. The products to cellular respiration are 6 carbon dioxide molecules, which we exhale, 6 water molecules, which we also exhale, and then ATP, which the whole reason cellular respiration happens is to make ATP, which is cellular energy for our cells. Our cells can't function without ATP. And so in eukaryotic cells, they have mitochondria that can do cellular respiration. In prokaryotic cells, it's a slightly different thing. They don't have mitochondria, so they can't do this exact same thing we're going to talk about. They do fermentation, which we'll talk about briefly at the end. So in a cell, you have cytoplasm, and then you have your outer membrane of your mitochondria. And then you have the inner membrane space, the space between the outer membrane and this cristae or inner membrane. And then in here you have what's called the matrix. And this is the inner membrane, which has folds on it, which we call cristae. So the folds have a special name called cristae. So inside of a mitochondria, you've probably seen it. Sometimes we draw them like this, where they're really folded. Mine's less folded, so I can draw pictures in it. But the reason that the membrane on the inside of the mitochondria is folded is because cellular respiration, part of it anyway, the most important part, has to happen across the cell membrane. So it happens right here. This is ATP synthase, and all three of these are proton pumps. And this is a reaction that happens, it's a final reaction that actually makes ATP. And it has to happen across a cell membrane because there's some passive transport and active transport involved that have to occur across a membrane, okay? So if we have more membrane, then there's more places that can do this specific chemical reaction. I only drew 2, but in a mitochondria, there could be thousands of these little ATP enzymes and proton pumps all over that can do this chemical reaction. So just for an example, if this is my mitochondria and this is my membrane, there's no way to get this piece of paper into this mitochondria this way. This is why a membrane is not flat, it's not straight, it doesn't work that way. If I were to fold, well, first off, I'm gonna put a bunch of dots on here, all the different places where this chemical reaction's happening on this cell membrane, or on this membrane inside the mitochondria. So if all of these little black dots I drew are these little ATP synthase enzymes. So if I need this to fit into my mitochondria, there's no way to get it in this way. The way you have to put it in is to wrinkle it up, fold it up like this. It's the exact same amount of surface area, it's just folded up. So all those little dots that represent the ATP synthase are still going to work and still do the chemical reaction. but they're folded and they fit inside of my mitochondria now. So that's just a little explanation of why surface area is important and why the cell membrane or the inner membrane inside of mitochondria has to be folded. Just makes more space for more chemical reactions to happen, for more ATP to be made. Okay, so outer membrane, inner membrane, the space between them, and then the space on the inside of the inner membrane is called the matrix. So we're actually going to talk about cellular respiration backwards. I'm going to start by explaining the last step, then the second step, then the first step. And it'll make sense when we talk about photosynthesis, because basically photosynthesis is this exact same thing, but backwards. So I'm teaching you photosynthesis and cellular respiration kind of at the same time because they're related, okay? But we're going to start with this. The first thing you need to know is that this thing is called ATP synthase. It's a protein that it's basically a protein channel, and protons, which when I draw protons, they're hydrogens, basically, hydrogen protons. They don't have electrons. It's just the one proton that comes with hydrogen because hydrogen's number one on the periodic table of elements. So when I draw protons, I'm just going to draw little plus signs to represent my hydrogen protons. So what happens is hydrogen protons go through from one side to the other, the ATP synthase protein channel. So there has to be a high concentration of protons on one side of the membrane and a low concentration of protons on the other side of the membrane, because what happens is, like all substances do using passive transport, they want to go from high concentration to low concentration through the ATP synthase channel. It's basically an open hole, like a protein channel. When protons go through the ATP synthase from high concentration to low concentration, it causes ATP synthase to spin. So let me take my cell membrane or my membrane out of here. This is now ATP synthase. Basically, if this was open on one side and open on the other, protons are going to go through and cause it to spin like this. And when it spins, ATP synthase, synthesis means to make something. ATP is what we're making. So ATP synthase is making ATP by putting together one molecule of ADP and a phosphate group. Every time ATP synthase is spinning, it's putting together molecules of ADP and phosphate. So if this was my ADP molecule and this was a phosphate group, when ATP synthase spins, it forces those two molecules together. And it keeps forcing them together every time it spins around. And it can only put those two things together when it's spinning. So it has to be spinning to make ATP, and protons have to be going from high concentration to low concentration to make it spin. Okay, so now we're going to get to the proton pumps. There's 3 proton pumps for every ATP synthase enzyme protein channel. And they pump protons from one side of the inner membrane to the other, because they have to make a high concentration of protons to flow back through using passive transport. So this part of the process is passive, going from high to low, but we have to force protons to maintain a high concentration by going from low to high. So these proton pumps pump protons against the concentration gradient from low concentration to high concentration, because there always has to be a high concentration of protons on one side, so they will flow through to low to make ATP synthase spin so that it will make ATP. So one thing you hopefully remember from transport is that active transport, going from low concentration to high concentration, takes some energy. So where we get this energy from is electrons. There's a process up here called the Krebs cycle, which we'll get to in a few minutes. It makes a couple different things. One of the things it makes is electrons, okay? These electrons make their way down to here where they are used to power our proton pumps. Each proton pump gets 2 electrons, and they get passed down the chain, powering the proton pumps to pump protons through until they get here. Now, this ATP synthase doesn't need electrons, and if the electrons are stuck here, then they've done their job and they don't continue to pump protons. So the electrons have to constantly be flowing through. There always has to be a constant stream of electrons getting passed down from one pump to the other. So there has to be something that comes here to collect electrons and take them away. This is what oxygen's job is. Oxygen's sole purpose, I see the video, you can't really see that very well. Let me try this again. The sole purpose of oxygen is to collect electrons from this electron transport chain. It takes these electrons, and for every 1 oxygen molecule, it can take 2 electrons. And it forms water. Because there's also protons in here, so it collects some electrons, 1 electron, and then 1 proton, and makes a hydrogen, basically. So for every 1 oxygen molecule, which this would actually be 2 molecules of water, because there's 2. For every 1 oxygen, you can get 2 hydrogens and make a water molecule. But the reason we breathe in oxygen is just to collect electrons from the electron transport chain in our mitochondria during cellular respiration. Oxygen is called an electron receptor. Its sole purpose in our bodies is to collect electrons from the end of the electron transport chain. Okay. So we've talked about ATP synthase. When it spins, it puts ADP and phosphate together to make ATP molecules. I didn't mention how many it makes. So for every 1 molecule of glucose and 1 O2 molecule, ATP synthase can make 34 to 36 ATP molecules, approximately, give or take. It's a range, basically. All right, so that's the last step of cellular respiration. We're going to move on to the next step up here. So this is called the Krebs cycle. So in the Krebs cycle, we're getting the products from the other process that happens in the cytoplasm. We'll get there in a second. But what basically comes in is 2 halves of glucose. 2 halves of glucose. We're going to call them 3 carbon molecules. They're also called pyruvate. But basically, half of glucose would be 3 carbons, 6 hydrogens, and 3 oxygens. That's what pyruvate is. It basically splits them exactly in half, and we're just going to call them pyruvate or 3 carbon molecules. But that happens in the cytoplasm. Where it comes from is that goes to the Krebs cycle and then gets broken down further into carbon dioxide, which is just a waste product. And where does it go? It goes over here. Also, where did our water come from? It comes from oxygen making water when it collects electrons. So our 3 carbon molecules get broken down into carbon dioxide and electrons, which we have here, and electron carriers, NAD. Yeah, NAD and plus. Once NAD plus picks up an electron, It's an electron carrier. Once it picks up an electron, it becomes NADH. Mostly, I just need you to know that NAD, anything that has NAD on it means it carries electrons, okay? So NAD plus carries electrons from the Krebs cycle to the electron transport chain. Because this is where we need the electrons to pump protons to high concentration. The Krebs cycle, while also making NAD and something else called FAD, and actually it's a electron carrier also. There's two different types of electron carriers. I think one of them carries more electrons than the other. You don't need to worry about that. Just know they both carry electrons. So Krebs cycle breaks down our 3 carbon molecules into NAD, FAD, CO2, and also makes 2 ATP molecules in the process. We don't need to worry about how. We just need to know that it does, because in our total, when we figure out how much ATP cellular respiration makes, it can make up to 38 ATP molecules. So we'll just add that in later. Okay. The last step, well, actually the first step, happens in the cytoplasm. I'm running out of space, so I'm going to erase this. The last step is called glycolysis. This is where we start with our glucose. When you eat, the carbohydrates in your food get broken down to basic monosaccharides like glucose. And glucose is called, glycolysis is called this because it breaks. Lysis means to break. And then glyco is like a prefix for glucose. This means to break glucose. So this process breaks glucose, which is a 6-carbon molecule like this kind of, basically. Breaks it in half into 2 pyruvate molecules, which are these 3 carbon molecules that I talked about over here. While it does this, glycolysis also makes 2 ATP. It has a net gain of 2 ATP. Okay, so most of our ATP comes from ATP synthase, where we make the most ATP, so like 34 ATP pretty much. Then we use our oxygen as an electron receptor in this step, which I forgot to circle, where we also make the water from our reaction. We start with glucose, and it breaks down into 2 3-carbon molecules, which go to the Krebs cycle, which happens inside the matrix of the mitochondria. This is important. So glycolysis happens in the cytoplasm. It doesn't actually happen in the mitochondria. Then the Krebs cycle happens in the matrix, which is inside the mitochondria. And then the electron transport chain occurs across the cell or the membrane of the cristae in the mitochondria. So from beginning to end, we have glycolysis. It breaks down glucose into 2 3-carbon molecules called pyruvate. Then they make it to the inside of the mitochondria. Don't worry about how. And the Krebs cycle breaks them down into NAD, FAD, and CO2, while also making 2 ATP molecules. Then these electrons, also from your food, get carried by NAD and FAD to the electron transport chain, where the electrons are used to pump protons through active transport across the membrane to form a high concentration of protons on one side of the membrane, so they can go through passive transport, like diffusion, facilitated diffusion actually, through this ATP synthase channel that will cause ATP synthase to spin. And when it's spinning, it puts together an ADP, and a phosphate to make ATP. To keep the electrons flowing, you have to have oxygen as an electron receptor to pick up the electrons and take them away, forming your waste product, water, when that happens. Okay, these are the steps of cellular respiration, the detailed steps. I hope you can see them. I will also put a filled out diagram on Canvas next week because I want you guys to do your own first, okay? But I will get you a clearer copy later. So do what you can with this. If you have any questions, come see me in tutorial during Zoom hours or email me. And I hope you have a great day.